everyone. I'm Vivian Bi from Ashland. It's my pleasure to be here today and discuss with you on how to overcome the challenges of polysoluble compounds through rational formulation design. Here is the outline of the presentation. I will go through different formulation approaches that are commonly used for polysoluble compounds. Then, I will focus on solid dispersion, which is one of the most effective and widely used solubilization technology. At the end of the presentation, I'm going to share with you Ashland's contract service capacity that can help pharmaceutical researchers to develop polysoluble compounds. The modern drug discovery process, such as rational drug design and high-throughput screening, are likely to generate compounds with higher molecular weight, higher lipophilicity, larger number of hydrogen bond donors and acceptors. All of this will contribute to the alarmingly high percentage of polysoluble compound. For compounds with high lipophilicity, lipid-based drug delivery systems and complexation using cyclodextrin might be very effective. For compounds with high crystal lattice energy, crystal structural modification, such as formation of amorphous solid dispersion, might be the best way to overcome the poor solubility. In this table, I'm showing several commonly used solubilization technologies. Some of the technologies, such as size reduction, the addition of super disintegrant and surfactant, can enhance dissolution rate, but have no or limited effect on drug solubility. Enabling technologies such as amorphous solid dispersion, drug complexation, and lipid-based drug delivery systems can increase drug solubility, therefore are more effective in many ways. Among them, solid dispersion probably have the broadest applicability. Therefore, my presentation today will focus on solid dispersion. Amorphous form is a thermodynamically unstable configuration of the molecules. Therefore, it has higher apparent solubility and dissolution rate compared to its crystalline form. Meanwhile, the thermal unstable configuration also bring about the stability issue, and crystallization tends to happen very readily and rapidly. Therefore, in most of the cases, the addition of polymeric carrier is necessary to stabilize amorphous material by reducing its mobility. In addition, polymer can also serve as crystallization inhibitor in aqueous media and help to maintain supersaturation of polysoluble compound in GI tract. Needless to say, the selection of right polymeric carriers is of critical importance to ensure good physical stability and dissolution performance of a solid dispersion formulation. Here is a list of typical polymeric solid dispersion carriers. The non-enteric polymers are not ionizable and can dissolve fast in both gastric and intestinal fluids. These polymers include Copovidone, Polymer, HPMC, and HPC. Enteric polymers has acidic functional groups, therefore are only soluble in intestinal environment. The ionized form of the polymers might be able to form colloidal species with drug in dissolution media, which can help to maintain supersaturation. Now let's take a look on the key considerations during polymer selection. It's not difficult to understand that the selection of solid dispersion carriers is very API dependent. For example, if the API is only absorbable in upper GI tract, then the selection of an enteric polymer which dissolves only at a high pH range might not work. Typically, the major things to look at during polymer selection are their physicochemical properties and their processability. Of course, other factors such as their safety, regulatory status, and compatibility with API should also be considered. From physicochemical property perspective, glass transition temperature, TG, and the interaction between polymer and the API are the two most important factors to consider. A high TG polymer can effectively reduce the mobility of amorphous API, therefore enhance its physical stability. The interaction between API and the polymer will impact the mobility, solubility, miscibility, and precipitation inhibition effect, and eventually will affect both the physical stability and dissolution performance of the solid dispersion. Here is a case study to demonstrate how polymer selection will impact the performance of Simvastatin solid dispersion. Solid dispersion at 25, 50, and 75% drug load were prepared with HPMCP, HPC, HPMC, different grades of Povidone and Copovidone. As illustrated in this table, after spray drying, all of the Povidone or Copovidone solid dispersions are amorphous. Well, when cellulosic polymer were used, trace crystallinity or phase separation were observed at 50% drug load. Povidone and Copovidone both have CO double bond and are strong hydrogen bond acceptors. They can form hydrogen bond with the hydroxy group in Simvastatin, which helped to form amorphous solid dispersion and maintain good physical stability. The hydrogen bond also explains why Povidone can significantly enhance the dissolution performance of Simvastatin, but cellulosic polymer couldn't. We've talked about the importance of physical-chemical property of the polymers. Now let's take a look at their processability. There are two major process technologies for solid dispersion preparation, spray drying and melt extrusion, and both have their advantages and limitations. There's no one technology that can meet the needs of all APIs. Normally, melt extrusion is preferred for simplicity and less environmental impact, while spray drying often can offer better mixing efficiency and low processing temperature. Therefore, it's more suitable to handle heat-sensitive APIs. For melt extrusion process, an ideal polymer should have good thermal stability, suitable TG, and melt viscosity. For spray drying, the polymer need to have decent solubility in one or more common organic solvents with the API, and the solution viscosity should not be too high to jeopardize the formation of droplets. What listed here in this table are the glass transition temperature of commonly used polymeric solid dispersion carriers. As mentioned before, the high TG polymers are desirable from physical stability perspective, but high TG polymers are also more difficult to process by melt extrusion. Some polymers, such as HPMC and high molecular weight Povidone, cannot be processed easily without plasticization. On the other hand, Povidone, Copovidone, low molecular weight Povidone, and some grade of the methacrylic acid copolymers can be extruded very easily. Here is an example to show that the melt viscosity of unextrudable Povidone K2932 can be reduced by the addition of plasticizers. Some of the plasticizers are more effective than the others, and Povidone K2932 becomes extrudable with the selection of the right plasticizers. What you are seeing here in this table is the solubility of commonly used solid dispersion carriers in organic solvents. For polymer with good solubility in different kinds of organic solvents, it will be much easier to find a common solvent with a poorly soluble API. In addition, good solubility of organic solvents allows feeding solution to have higher solid concentration, which can enhance the throughput of spray drying. One example of such a good solubility polymer is Copovidone. On the other hand, A polymer with poor solubility in most of the organic solvents could result in less process flexibility and lower throughput. Here is a list of marketed solid dispersion products. If you take a look at the polymers that are used in these products, they are Polydone, Copovidone, HPMC, and HPMCS in most of the cases. The reason that these polymers are in the commercial product is that they have desirable overall profile as solid dispersion carrier, including physical chemical properties, processability, and safety property. Now, I'd like to use one case study to demonstrate how process technology or process condition can impact the performance of solid dispersion. The model compound here is Piroxicam. It has melting point of around 200 degrees, a TG of at 62 degrees, and a water solubility of 2.3 microgram per milliliter. Solid dispersions were prepared at 30 and 40% drug load with Plasdone at 630 Copovidone using both spray drying and melt extrusion. During spray drying, all process conditions are fixed except for the solid concentration in feeding solution. The solution concentration varies from 5 to 7.5 to 10%. For the melt extrusion process, 2 screw speed, 500 RPM and 700 RPM were evaluated. In addition, the impact of screw configuration were evaluated. As shown in this screw configuration diagram, on one screw there were 2 kneading blocks. On the other screw, there were 4 kneading blocks. The bio temperature are shown in the middle of the slide. The highest process temperature used here is 140 degrees, which is below the melting temperature of the API. Here are the pictures of the resulted extruded. The top 2 rows are the results of 30% drug load samples, and the bottom 2 rows are those of the 40% drug load samples. At 30% drug load, the extruded are transparent, while at 40% drug load, the extruded lost transparency. The loss of transparency in the case can be used as the initial indicator of the residue crystallinity. Now let's take a look at the X-ray powder diffraction patterns of the solid dispersion at 30% drug load right after the sample preparation. The left-hand side is the result of spray-dried solid dispersion. The samples are amorphous no matter which solid concentration level was in the feeding solution. The right-hand side is the result of melt extruded solid dispersions. At screw speed of 700 RPM, both samples are amorphous. At screw speed of 500 RPM, only sample proceeded with 4 kneading blocks generated amorphous solid dispersion. It is not hard to understand that at high screw speed and with more kneading blocks, the sample can go through more shear thinning, therefore can help to enhance the mixing efficiency. Here are the dissolution profile of solid dispersion at 30% drug load. For the spray-dried solid dispersion, significant improvement in dissolution was observed comparing with crystalline API, and the solid concentration in feeding solution also have a big impact in the dissolution performance. In this case, the higher solid load in feeding solution generated samples with longer supersaturation time period. During the spray drying process, low molecular weight components such as solvents and API could have greater molecular mobility during droplet formation. Therefore, a higher concentration of API could move to the outside of spray-dried particle. As seen in this study, a high solution concentration may decrease this molecular mobility during the spray drying process and generate more evenly distributed solid dispersion with better dissolution performance. On the other hand, the extruded samples were also able to enhance the dissolution of Piroxicam significantly, although they were not able to maintain supersaturation for a very long time. In addition, the dissolution performance of all extruded are very similar regardless which screw speed or screw configuration was used. Now let's take a look at the extra powder diffraction pattern of 40% drug load samples. Once again, all the spray jet samples are amorphous, no matter what is the solid concentration level in feeding solution. For the mild extruded samples, all have shown partial crystallinity, which suggests that at this drug load, the mixing energy could not fully overcome the crystal lattice energy of the API. Here are the dissolution profiles of the 40% drug load solid dispersion. Once again, spray-dried solid dispersion produced with high solution concentration showed lower crystallization tendency and better maintenance of supersaturation. For the mild extruded samples, dissolution performance was impacted by process conditions. The sample generated under higher screw speed with more kneading blocks showed the most significant dissolution enhancement. For this case study, it is obvious that drug load, process technology, and process conditions can have significant impact on both the dissolution and physical stability of the resulting solid dispersion product, and therefore should be optimized to achieve desirable product performance. So hopefully from this presentation, we will understand that the solid dispersion technology is a very effective technology for the bioavailability enhancement of poorly water-soluble compound. When we select a polymer for solid dispersion, we should consider both physical-chemical properties as well as the processability. A holistic view from API, Excipients, material science, and process perspectives is very critical in the design of rational formulation of poorly soluble compound. Before wrapping up the presentation, I'd like to take a few minutes to walk you through Ashland's Excipients and technical service on solubilization. To those who are not familiar with our relatively new identity yet, Ashland acquired Hercules in year 2008 and ISP in 2011 and become one of the top specialty chemical company with a very broad portfolio of pharmaceutical excipients. For drug solubilization, we have vinylpyrrolidone polymers such as PVP and Copovidone. For cellulosic polymers, we have Benicell HPMC, Clucell HPC. And I also want to let everybody know that we have a new product, which is HPMCES, and we are going to have the product launch later this year. In addition to the polymeric carriers for solid dispersion, we also have cyclodextrins, including alpha, beta, and gamma cyclodextrins, as well as 2 grades of HP beta CD. With our formulation experience, material science expertise, and the know-how on process technology, we can provide contract service to pharmaceutical clients from proof of concept study to formulation screening, all the way to CGMP manufacture. This slide here highlights Ashland's expertise, experience, and know-how in solid dispersion development. In the past years, we have formulated around 100 compounds using different solubilization technologies. Our work has led to 9 patents filing and 2 proprietary process methods. In solubilization area, Ashland has a unique combination of a broad portfolio of excipients and contract service capacity. The picture here shows the result of a client project in which we were able to successfully convert a 16 capsule dosage form to a single tablet dosage form. And we were able to deliver the product from the formulation screening all the way to clinical supply manufacture within 4 months time period. We would be very happy to work with you in speeding up the development of your polysoluble compound. Thank you so much for your attention, and I'd be happy to take any of your questions. Thank you. Now I got several questions from the audience already. The first question is around HPM-CAS. The question says, for your new HPM-CAS product, when are you going to have the sample available, and whom should we contact for samples? Actually, we're going to have the H, M, and L grade of HPM-CAS in June this year. So we're going to have a product launch. And all the customers, if you're interested, you can contact the person who are listed at the right-hand side of the window. We have 3 persons listed there. You can send email to either of them and get the sample. And if you can contact us earlier, and we can try to arrange a sample for you as a higher priority. So thank you for that question. I do have another question regarding the case study of Piroxicam solid dispersion. Is that higher solid load of the feeding solution result in better dissolution behavior? Does that mean one should always use a high solid load feeding solution for better quality of the product? That's a very good question, and I would say the answer is really case by case. In some of the cases, such as the one that I shown here at today's presentation, actually we can see the API, the drug, migrate during the spray drying, and the higher the solid load, the more viscous the spring solution, feeding solution will be, and in that case, you get less migration of the API, and you get a more evenly distributed solid dispersion particles, which result in better dissolution. But in some other cases, the migration of the API is not as significant, and you won't get the same kind of a result. And another thing one should consider is that when you have a very high solid load in the feeding solution, most likely the viscosity could cause some spray drying issue. You can get a droplet without very good shape, and then the particle will not be as round. You could have problems such bearding. So it's a fine balance between the performance and also the processability. Another question I got here is that is your contract service facility located in US? Do you use your own excipient only for the formulation screening of solid dispersion? Actually, our contract service facility is located in US. It's in Columbia, Maryland, and for the formulation screening and formulation development, we use all kind of excipient, including vinyl pyrrolidone polymer, cellulosic polymer, and methacrylates. So we use all sort of polymers. But on the other hand, since Ashland offer very broad range of excipients, we offer vinyl pyrrolidone polymers and cellulosic polymers, and the majority of the excipient we are using, we also provide the excipient, and so that's for the excipients that we don't make by ourselves. We also use them for customers' formulation screening. I do have another question here, and saying, have you encountered any cases that solid dispersion cannot solve the solubility problem. Actually, we very rarely see any cases that solid dispersion cannot enhance the solubility or dissolution rate of some certain API. We do sometimes see very challenging case though, and I want to give you an example. Actually, we have one compound that we encountered just recently that only at 5% drug load we are able to get a stable solid dispersion formulation, and that solid dispersion formulation cannot maintain supersaturation in GI tract or in dissolution media. So what we did was we used a combination of water-soluble polymer and water-insoluble polymer, and through the dual mechanism to prevent the crystallization, we were able to achieve a good supersaturation in dissolution media. So that's a very extreme case that we have explored many different approaches and to make the solid dispersion work. So in another word, I think when we have a solid dispersion formulation, the challenges that we are facing could vary a lot depending on which compound we are working with, and in most of the case we are able to enhance the dissolution or the solubility of the compound. Any other questions? Okay, here is another question talking about, you mentioned about the importance of the processability of excipient. At which development stage should processability be considered? This is another good question. I think processability should be considered as early as possible during the formulation development. For example, if you have a compound with a very high melting point, and you are also thinking about the relatively high drug load, and in that case, probably you will have an idea that hot melt extrusion might not be a good option for process. In that case, you might want to focus on the solubility of the compound in different organic solvents, and also select some excipient that have a good solubility in organic solvents, so that you can spray dry the compound you have. So the earlier you consider the processability, the more cost efficient and you can save more time during the drug development. I have another question here, and asking about, do you offer services for tablet or other final dosage forms? Yes, for our contract service part, we do offer service for the final dosage form. We have formulated tablets, we have formulated capsules, and those tablets can be controlled release, can be IR, and so we develop the formulation dosage form to supply to the clinical study and late stage clinical study. So we do handle all the final dosage form development here. Here is another question asking about, why did 40% drug load solid dispersion keep supersaturation keep supersaturation better than 30% drug load solid dispersion? I think this question, probably if you go back to the slide, which I think you can see the re-recording later on, actually 30% drug load and 40% drug load have similar effect in this case study. So if you have, this is a question from Koji, and if you have any further question, I think we can communicate through email. And I think soon we need to wrap up because the time is up. For any of you have further questions, you're very welcome to send email to me directly. Thank you so much for your attention.